I think we didn't realize that we were on an exponential when we started sequencing. I got introduced to it through RNA sequencing. There wasn't DNA sequencing, and then Wally Gilbert um, was my mentor as a graduate student, and he invented it. His team in 1977 independently. Fred Sanger published a paper that same year. It took a little longer for the Sanger method to get implemented, but what happened was very quickly we got to a point where we were talking about doing a whole genome, mainly at the Department of Energy's bequest in 1984. They asked a harder problem, which was how do you estimate mutation rates as a consequence of energy, and we felt, a handful or maybe 10 scientists in what would later be called genomics said, well, we can't do that, but what we might be able to do is get one genome, and a reference genome, and then that consolation prize was big enough that Charles DeLisi at the Department of Energy just started writing checks. I mean, he didn't wait for an act of Congress or anything, just had money for this kind of R&D because of health effects. And then it took about 3 years. So my lab, I was transitioning from postdoc to professor, and my lab got one of the first 2 Genome Project grants. And then the NIH, it took about 3 years before the NIH got involved, but they got involved in a big way because they felt they were more appropriate vehicle for anything health related than the Department of Energy. And they did it kind of in a teamwork with maybe 30% DOE and 70% NIH in the United States component, plus lots of international collaboration, really starting in 1990 with a 15-year goal. There was a lot of talk of cutting corners at the beginning. I didn't necessarily call it that, but there was a lot of people trying to do 1x coverage, meaning doing every base pair exactly, reading it exactly once. And I didn't like most of these corner cutting things, but I was the most junior member of the project from the beginning, didn't have a lot of sway. I also felt that we should put technology development up front because that could reduce the price, and then we could do a lot more than one genome for a lot less than $3 billion. But as soon as some of the senior members of the visionary team, like Jim Watson, who came in later, started representing, drumming up support in Congress, it became evident that we would have $3 billion, and then the motivation for bringing the price down disappeared for a few years, a decade. Then instead of some of the corner cutting was to not worry too much about repetitive sequences, which in the case of the fruit fly was about half the genome. It doesn't matter. And at one point they were going to just do the coding regions, which is 1%. Turns out we still haven't identified the 1% coding regions. That would not have been a shortcut. So a lot of these shortcuts were really ill-conceived. But fortunately, we did get a decent 92% of the genome and declared victory. I want to make sure we've got that before we go on to writing genomes as a whole, another topic.
Do we need more reference genomes? And what are your expectations of finding new tools elsewhere in the evolutionary tree?
Well, so yes, we certainly need more genomes. It's not just the reference, it's the population variation that's important. We want to, the variation is at least as important as the reference, and it helps us make sure we've got a good reference. So you can call that the reference. It's growing recognition that we can represent the reference as a diversity. We are finding tools in the genomes. So one of the nuances that developed, the first kind of recommendations for maybe the 1984, 1985, 1986 was the human genome, as if there were one and as if there weren't any other genomes. And I kept advocating for genome comparisons, because when you compare two genomes, that's almost as good as an experiment, but it gives you a richer formulation for exploration. And we have part of that genome comparison has resulted in new tool discovery. And so it's kind of a positive feedback loop. You sequence some genomes, you find some tools, use those to read and write genomes, find some more tools, and so on. I don't know where that ends, but I do think that synthetic biology is probably ultimately unlimited, while the diversity on Earth, even though it's vast, is limited, more limited. Almost by definition, we can explore more than currently exists, at least initially in narrow corridors where we're looking at specific tool building, ecosystem restoration, and medical consequences. I think there's a rich field of let's say you had one book and that's the only book you had. You could read it and reread it and reread it, and you'd keep learning more and more. But as soon as you start writing books, now you got millions of them. So that's how I think of the synthetic biology or writing of genomes. I should mention that we don't even have the full instruction book of any human being yet. We declared victory in 2001 on a kind of a rough draft of 92%. Actually, it was considered the final draft of a rough draft in 2001. It was final draft in 2004, but it was still haploid, meaning it was just one genome, while essentially all of us are diploid, inheritance from mother and father, except for our gametes. So the sequence we have, the one human genome that we have, is not of a gamete; it's of a strange haploid cell. But that's not the big barrier to understanding. The big barrier is, as you say, is in order to understand how something works, and also in order to develop new technologies, you need to be able to write and edit and alter.